With that, I'll go right into the first presentation, which is entitled Clinical Trial Design in Hemophilia. And again, this is the work of a project group, and I will be presenting this on behalf of the project group entitled Clinical Trial Design for Hemophilia, and this project group is reporting to the Factor VIII, Factor IX subcommittee of the ISTH. I have no disclosures, so I'll go right into explaining a little bit about the mandate for this project group, which began in February of 2011. And that mandate came from issues that were both pragmatic and regulatory. From a pragmatic standpoint, we were faced with a time in which multiple products were entering into pre-registration, pre-licensure trials all at the same time. Simultaneously, we have the situation in which the availability of hemophilia participants in this trial was limited, given both the fact that, again, hemophilia is a rare disease and the current regulatory requirements for subject number, trial design, trial endpoints, etc. And although we were talking about hemophilia, and we're going to talk about hemophilia A and B with respect to the current work that's ongoing in this project group, certainly we well recognize that this product is further amplified in even rarer disorders. The other pragmatic consideration was that if you looked at all the stakeholders, industry, regulators, clinicians, patients, all were demanding trial simplification, but without a compromise in safety and efficacy assessment. From a regulatory standpoint, the issues related to the variability in the requirements between primarily the European and the American regulatory agencies with respect to both pre- and post-registration safety and efficacy assessments for licensing clotting factor concentrates. And although the underlying philosophy of safety and efficacy of biologics was very well appreciated, the scientific basis for some of these regulatory requirements were unclear to many. And most agreed that the value of discussing and harmonizing regulatory requirements was certainly very important in moving forward. So with those issues in mind, the aim of the project group was to determine optimal prospective pre-licensure as well as post-licensure observational trial designs for new clotting factor concentrates for hemophilia, taking into consideration potentials for harmonization of safety and efficacy data required by regulators for project registration, the anticipated study population that was going to be available for all of these trials, and the potential for introducing innovative clinical trial designs that were suitable for rare diseases such as hemophilia. So the group that was brought together and that I have the good fortune of chairing included members of both regulatory agencies, as well as the individuals who were inspired to begin this project group, former and current chairs of the Factor VIII, Factor IX subcommittee. We asked some individuals with expertise in epidemiology and clinical trial design to join us, and particularly those individuals like John Scott from the FDA who have experience in small clinical trial design. And because a lot of the clinical design issues dealt with product immunogenicity and neoantigenicity, we invited an immunologist to be a member of this group. As I said, the project group began in February of 2011. We've had roughly 16 meetings since its inception. We meet roughly on a monthly basis, and the final report from this committee is due in June or July of next year. So, in thinking about how to approach this, we wanted to envision the potential for alternative clinical trial design and statistical modeling and its application to 5 different sort of types of trials, largely PTP with some consideration for PUP trials, but largely PTP trials that dealt with factor VIII products, factor IX products, and bypassing agents. And among those products, we were very interested in looking at molecules that were considered to be biosimilars, Me Too products, unmodified products, as well as those products that were considered to be novel, largely increased potency and extended half-life products. And there were two objectives. We sort of decided on two objectives. The first was to refine or develop, if there weren't any, consensus clinical outcome endpoint definitions that included definitions of clinical severity, prophylaxis, inhibitors, hemorrhage, response to treatment, and then take those concepts, take those definitions into an optimized clinical trial design that took into account and really reevaluated the numbers and types of subjects that were needed both for safety and efficacy trials as well as surgery trials, and to also look at the current requirements for study duration, exposure day requirements, and assay methodology. With respect to both those activities, there were sort of the group divided into two subgroups, and the clinical trial design and statistical modeling subgroup, the members of which you see there, were tasked with two primary issues. And I'd like to really recognize the work done by Nisha Jain at the FDA in terms of her look-back project at post-registration inhibitor development, and John Scott, who led us through basically a mini course in small clinical trial design, and Sébastien Lacroix-Damas, who helped us understand what we mean when we're talking about neoantigenicity. So largely through their efforts, there is an attempt going on in this subgroup to re-examine PTP immunogenicity clinical trial requirements for both factor VIII and factor IX biosimilars, and the potential of reapproaching this by looking at priors, looking at currently available data, and the potential to apply Bayesian as well as adaptive design strategies to optimizing these clinical trials, particularly pre-registration trials. The applicability of priors for novel products is something that is still being examined, and we're certainly waiting for some data to appear that might allow us to approach novel products in somewhat of the same way. And then the other task was to think about how we can introduce mechanistic studies looking at alternative ways of looking at early immunogenicity and neoantigenicity in order to eventually be able to implement early detection assays that we can't use right now. The clinical endpoints group was basically trying to pull together all the activities going on in this area through liaisons with our definitions project group as well as the global assay working group initiated by the NIH to develop more precise consensus definitions for objective clinical efficacy endpoints that could be incorporated into future trials, and to really go to all of the stakeholders to really see if we could develop a consensus model. Now, who are those stakeholders? Well, a lot of the stakeholders were represented within the committee itself, but we also did a tremendous amount of work working with the definitions project group, the work of which you're going to hear in the next presentation. And we've gone out several times to the clinical and patient communities through presentations at the WFH Global Forum, the ISTH in Kyoto, and now this presentation. And these presentations are largely to get some of the clinical stakeholder input into this process. But we also wanted to go to our industry representatives to ensure that we had their ideas about optimizing this process. So to that end, the industry was polled through a survey. And the goal of the survey was to help us better understand from an industry perspective the relative burden of the current regulatory requirements, as well as the potential alternative approaches to maybe getting around those requirements or modifying those requirements for both pre- and post-registration trials, for Factor VIII and IX products initially, both novel and biosimilar or Me-Too products. And what we asked for comment on required subject number, inclusion criteria, PK requirements, inhibitor testing, clinical endpoints, and pediatric subject requirements. We heard from 10 industry representatives, and actually industry was asked to do this in relatively short order, and I must thank all the industry participants for their very honest and very thoughtful feedback. Among those, there are 3 that currently market only plasma-derived products, 3 that have only Me-Too products in development currently, and 4 that have novel products in the pipeline. What I'm going to present next actually is a preliminary evaluation of those comments. I think we need to look through them further. But what I'd like to do is to say 3 things about them in general. That some concerns were somewhat variable, industry to industry, and that the points that I'm going to highlight in this presentation are views that were the most frequently expressed. And that overall, there was somewhat more regulatory satisfaction with respect to the licensing of Factor IX products than for Factor VIII. So with that in mind, I'd like to go through some of the specific feedback. So the first question that we asked was, what, in your opinion, were the regulatory barriers to product registration? And please give us some idea of potential solutions. So what we got back was some concern about the current definitions of an acceptable inhibitor risk, as well as the subject number targets to evaluate acceptable inhibitor risk, and the thought that maybe these were not always as evidence-based as they could be. Some potential solutions included the application of Bayesian statistical approaches to using prior inhibitor data distribution, although it was acknowledged that this might have much more limited applicability to novel products. And some of the feedback we got back was about really thinking about the somewhat arbitrary upper bound of the 95% confidence interval for inhibitor incidence of 6.8% for PTP studies, and the thought that maybe this number might not be quite as applicable as it could be when the incidence of inhibitors in PTPs is 1 to 2%. The other feedback we got is, in terms of regulatory barriers, are the current definitions and required numbers for PUPs, PTPs, and exposure days, the numbers that are required and definitions required for licensing trials. And the feeling that these requirements were indeed hampering accrual and adding years to registration, particularly for novel products. Some of the feedback that we got in terms of alternatives was to potentially think about redefining PUPs to include what we used to call minimally treated patients, pediatric patients with 1 or 2 prior exposure days, and reconciling the definition of PTP, which is quite different if you're looking at a pediatric PTP patient, an adult PTP patient, and potentially reconciling the definition of PTPs of all ages. With respect to exposure days, there was a lot of concern from those industries that are going into pre-registration trials with enhanced half-life products, and that 50 exposure days, as currently defined, might lead to trials that have additional years to registration, and suggested that maybe we need to apply the time exposed concept used to define an exposure day equivalent in prophylaxis trials. Other comments with respect to this question included that particularly those companies involved in designing trials for novel molecules that have sort of evolving clinical indications, that there was some difficulty in basing these on older regulatory paradigms for clinical indication and product labeling. There was certainly a lot of comment about poorly defined and non-harmonized clinical endpoints that were very difficult to adapt to non-homogeneous populations. And the suggestions with respect to this included the need for more regulatory innovation and adaptation to novel product requirements, some regulatory consensus on some points that included clinical and laboratory endpoints and definitions, particularly with respect to phase 3 trials, both treatment and prophylaxis trials. And also some consideration be given to PK with factor levels as a surrogate for pre-licensure efficacy, as the FDA is currently doing for rare orphan drugs for rare diseases. Now, the second question was, what would be the greatest benefit? Where would industry benefit most from EMA FDA harmonization? And there are two areas. The first is pediatric investigative requirements. And the comments that we received were that the lack of FDA and EMA harmonization on all aspects of pre-licensure studies with respect to pediatric requirements poses a significant obstacle to future product development, and I quote, and that EMA and FDA harmonization of pediatric study requirements was absolutely needed, with some comment that as the FDA has become more flexible, the EMA's pediatric investigative plan has become more rigid. And these were the concerns, that these requirements significantly delay adult access to new products, and that the need to have a pediatric investigative plan before adult studies are done really limit the use of adolescent and adult data in informing pediatric studies. And certainly those companies that have plasma-derived products felt these requirements to be particularly onerous. And so the recommendations were that consideration should be given to distinguishing between, when we're talking about pediatric subjects pre-registration, maybe distinguishing between pediatric PTPs and PUPs, and also rethinking the ratios of under 6 to 12 age patients, which are currently 1-to-1. And also thinking about decreasing the pediatric subject number if the PK results look like they're similar with those of adults and adolescents. Also, with respect to the EMA requirements, maybe beginning the pediatric investigative plan pre-registration but completing it afterwards, post-registration. And of course, we got some comments about collecting all pediatric data post-registration. The second area where the industry felt that they could most benefit from harmonization was with respect to pharmacokinetic requirements, including harmonization of time points and comparators, particularly in children, designing crossover studies with a sufficient number of PK studies to demonstrate bioequivalence between comparators and new products. Harmonization of potency assignment in assays was a big one, and really a better definition for minimally protective factor trough level for prophylaxis studies was also a comment. And of course, guidance for PK studies for the enhanced half-life products was sought. The third question we asked was, what are the perspectives on optimizing safety and neoantigenicity monitoring? And certainly all respondents responded that safety and neoantigenicity was a high priority. The majority thought that, however, shorter PTP neoantigenicity trials supplemented by rigorous prospective post-registration data collection in the real-world setting was certainly a way to go to optimize pre-licensure trials. However, there was a minority view that felt that most of the data should still be collected pre-registration because of the complications in trying to understand post-registration data, particularly due to product switches. And for novel products, again, the current definition of exposure day was a problem, and more rigorous post-registration monitoring was considered to be an alternative. Now, with respect to the same question on clinical efficacy and effectiveness monitoring, some of the suggestions were about harmonized lab assays, clinical endpoints, using PK as a surrogate for efficacy, revisiting the requirement for so many surgeries, revisiting the need for an on-demand control arm in prophylaxis studies, and really shifting most of the product efficacy to really product effectiveness studies in the post-licensure phase. Additional comments: the need for central IRBs because of the regulatory issues, even the current requirements post-registration for 100 exposure days, and really looking at the national registries that are being developed and wondering whether we can shift some of the data collection to harmonized endpoints within these national registries. So with that, the group's thoughts so far are indeed that although safety and efficacy are important, that safety is the greater design challenge, and that we really are going to have to live with inhibitor assays right now as we consider our recommendations, because there are no alternatives, although mechanistic studies need to be considered, and that we're still looking at whether unique solutions are going to be needed for each of the different products and product types. And we definitely need to look at the complementary role of prospective observational post-registration studies in optimizing this. Our future plan of action is to complete these ongoing activities with all the stakeholder input that we can get, and to begin to formulate our recommendations that will be presented in the Factor VIII, Factor IX subcommittee in June, July, and to continue to engage all of you in the feedback on this issue. So we look forward to some spirited discussion during the discussion phase. So with that, I'll end, and thank you very much.